Welcome back to another edition of the Worm Podcast.
And we are bringing some serious energy today.
We absolutely are, because we're tackling a subject that, let's be honest, obsesses pretty much all of us.
The quest to live longer.
But we aren't looking at Silicon Valley biohackers. We are not looking at cryogenics. We are looking at a microscopic nematode.
That's right, the humble C. elegans. The unsung hero of biology and maybe, just maybe, the creature that holds the manual for healthy aging.
Okay, before we unzip the secrets of the universe from this tiny, transparent worm, I have a quick favor to ask you.
Please do.
Please, right now, just hit that subscribe or follow button on whatever podcast player you're using. We are digging up some truly mind-bending research, and we do not want you to miss what's coming next.
Absolutely. It really helps us out, and you definitely won't want to miss what we have in the pipeline.
Okay, let's set the scene. We are diving into a paper published in Nature Communications back in July 2025.
And it's titled Immunometabolic Stress Responses Control Longevity from Mitochondrial Translation Inhibition in C. elegans.
It is a mouthful.
Oh, yeah. But the implications are just massive.
And look, we've known for decades that if you mess with the mitochondria, the powerhouse of the cell, right? Everyone remembers that from high school biology.
Right.
You can weirdly make organisms live longer.
Yeah.
That part is established science. But this paper, it basically says everything you thought you knew about why that happens is wrong.
It really pulls the rug out from under a major theory. For a long time, we thought longevity was all about repair. You know, you break something, the body fixes it, and you come out stronger.
Right.
This study suggests it's not about repair at all. It's about a complex immunometabolic dance. It's about fighting off invaders and, get this, changing the actual composition of your body fat.
It connects the immune system, metabolism, and aging in a way I have not seen before. But to appreciate the twist, we have to understand the original plot.
We need to talk about the mitochondria paradox.
The paradox, right. This is the baseline for all of this.
In almost every model organism we study, I'm talking worms, fruit flies, mice, if you inhibit mitochondrial translation, the organism lives longer.
And when we say mitochondrial translation, just so everyone's on the same page, we're not talking about translating languages here.
No, we're talking about protein synthesis.
Yeah.
Mitochondria have their own DNA, their own ribosomes. They build some of the proteins they need to function.
Okay.
So if you use genetic tools or certain antibiotics like doxycycline to slow that production line down just a little bit, you get this lifespan extension.
Which is the paradox. You inhibit the engine of the cell, you make it less efficient, and somehow the car runs for another 100,000 miles.
Exactly. And it's not just lifespan, it's health span. These animals, they move better, they are more active, and in mouse models, this specific stress can help delay conditions like Alzheimer's.
So, of course, scientists wanted to know why. And for years, the reigning champion of theories was something called the UPRMT.
The mitochondrial unfolded protein response.
Try saying that 5 times fast.
The concept itself is actually very elegant. The idea was that when you stress the mitochondria, by slowing down that protein production, you end up with an imbalance. You get these unfolded or, messy proteins accumulating.
Which is bad.
Usually, yes. But the cell senses this proteotoxic stress, and it sounds an alarm. That alarm recruits a cleanup crew chaperone proteins, proteases, to come in and fix the mess.
Okay.
And the theory was that this cleanup crew didn't just fix the immediate problem, they sort of spruced up the whole cell.
So it's like calling a plumber to fix a leak and he ends up renovating your entire kitchen while he's there?
That is the perfect analogy. The thought was, the stress triggers the UPRMT cleanup, the cell gets cleaner, more efficient, and therefore you live longer. Case closed.
But science is rarely case closed, is it?
Rarely. And this is where our authors, Hu and Help Cooper, step in. They noticed something that just didn't fit. They found instances where that UPRMT alarm was ringing, the cleanup crew was there, getting paid overtime.
But the worms didn't live longer.
Exactly. And conversely, they found ways to extend lifespan where the cleanup crew wasn't really the main factor.
So the correlation just...
It crumbled.
Hmm.
I mean, they realized the UPRMT might just be a side effect. It's the smoke from the fire, but it's not the water putting the fire out.
That is a huge distinction. So if the UPRMT isn't the hero, who is? Because something is clearly keeping these worms alive.
That's the mystery they set out to solve. They needed to find the real driver.
And they found it. A gene with the catchy name C32E8.9.
Classic nematode nomenclature. Doesn't exactly roll off the tongue.
I think I'll just stick to calling it the key for a moment. But how did they find this one specific gene out of thousands? It feels like looking for a needle in a haystack.
A haystack made of needles. They used a really smart evolutionary approach. They compared data from two different species.
Okay.
They took mice treated with doxycycline, which we know inhibits mitochondrial translation, and then worms treated with RNA interference to do the same thing.
So they're looking for the Venn diagram overlap. What happens in a stressed mouse and a stressed worm?
Exactly. They were hunting for a conserved mechanism. If this longevity effect works across species, the gene responsible should probably be present in both. And C32E8.9 just popped up as a top candidate.
And do we humans have this gene, or is this
Well,
just a worm
But
thing?
we do. Our version, the ortholy, is called ECHDC1. It stands for ethylmalonyl-CoA decarboxylase 1.
Okay, that makes C32E8.9 sound pretty catchy by comparison.
Fair point. In humans, we've known ECHDC1 is involved in fatty acid metabolism. It helps prevent the buildup of certain toxic metabolites. But until this study, no one had really linked it directly to this longevity mechanism.
So they have their suspect, C32E8.9. How do they prove it's actually responsible?
They did the gold standard of genetics,
Yeah.
the knockout experiment. They set up the conditions for longevity. They stressed the worm's mitochondria using MRPS5 RNAi.
So these worms should live longer. They're getting the treatment.
Right. And normally they do. They live longer, they wiggle faster, they even resist paralysis in Alzheimer's models. But then in a specific group, the researchers just deleted the C32E8.9 gene.
They pulled the plug.
And the result was stark. The life extension just vanished. The worms went back to a normal, shorter lifespan. All the health benefits gone.
But wait, what about the cleanup crew? The UPRMT, was that alarm still going off?
That is the kicker. They measured a marker for it called HSP6. The alarm was still ringing, loud and clear. The cleanup crew was deployed. But without C32E8.9, it didn't matter.
That proves it. The cleanup crew is there, but it's powerless without this gene.
Precisely. It completely decouples the stress response from the actual longevity benefit. It shows C32E8.9 is the necessary component.
Okay, so we have a new hero, but what does this gene actually do? You said it's metabolic, but the paper's title says immunometabolic. Where does the immune system come in?
This is where the story gets really complex and fascinating. The researchers did a transcriptome analysis, basically looking at all the genes being turned on and off.
A snapshot of the cell's activity.
And when they knocked out C32E8.9, they saw that all the beneficial gene patterns that are usually triggered by mitochondrial stress were reversed. And when they looked closely at which pathways were affected, it pointed straight to immunity.
Specifically, the TGF-beta pathway.
Yes, and TGF-beta is a huge deal in cell signaling. It controls everything from cell growth to immune responses in humans, too.
So in this context, it's acting as a defense coordinator.
It is. The study identified a partner gene called SMA4. It's a transcription factor in that same pathway.
So let me see if I can trace the chain of command here. You stress the mitochondria. That stress signal hits C32E8.9. This gene then ramps up SMA4, and SMA4 turns on the immune system.
That is the working model, and they proved it's causal. If you knock down SMA4, you lose the longevity benefit, just like you did with C32E8.9.
But is this just cellular immunity, or are these worms actually better at fighting off disease? Like, if they get an infection, do they survive?
They tested exactly that with a slow-killing assay.
Sounds ominous.
It is for the worms. They expose them to a pathogen called Pseudomonas aeruginosa. It's a nasty bacteria.
Right.
Usually, worms with that mitochondrial stress, the long-lived ones, they're like super soldiers. They resist the infection much better than normal worms.
Because their immune system is pre-activated?
Exactly. They are in a state of high alert, but, and you can guess what's coming,
If you remove C32E8.9 or SMA4,
that super soldier status disappears. They die from the infection just as fast as the control group.
so the longevity isn't just about not aging in a vacuum. It's about not dying from biological attacks.
Yes, but there was another twist that I absolutely loved. It wasn't just physiological, it was behavioral. They ran a food avoidance assay.
Food avoidance, like
Right.
recognizing a bad buffet.
More like recognizing poison. C. elegans feed on bacteria. But if the bacteria are pathogenic, if they make the worms sick, smart, worms will stop eating and physically move away,
This salad tastes like death. I'm leaving.
basically. Now, standard worms are okay at this. But the worms with the mitochondrial stress treatment, they were hyper-aware, They recognized the danger faster and avoided the bad food more effectively.
That is incredible. So the stress makes them smarter.
It enhances their survival instincts.
Yeah.
But again, take away C32E8.9 and they lose that edge. They just stay on the pathogenic lawn and get sick.
Wow. This really changes how I view aging. We usually think of it as just, falling apart.
Right.
But this suggests that longevity is an active program. It's a state of heightened readiness.
That's a key insight. The mitochondrial stress acts as a warning signal. It tells the body, something's wrong with the energy supply, we might be under attack, prepare defenses, and C32E8.9 is the general that mobilizes the troops.
Okay, so we have the immuno part down, but we still need to talk about the metabolic part.
The lipid shift.
Yeah.
This brings us back to what C32E8.9 and its human cousin ECHDC1 actually do enzymatically. They process fats.
And when we say fats, we aren't just talking about, stored calories.
No, in biology, lipids are everything. They are energy storage, yes, but they're also the structural walls of your cells and key signaling molecules. The researchers did a lipidomics deep dive, analyzing every fat molecule in the worm.
And what did they find?
A very specific shift. When mitochondrial translation is inhibited, when the worms are in that long life mode, they stop making short, saturated fats.
Saturated fats, usually considered the storage fat.
Broadly speaking, yes. Chemically simple. But instead, the worm started pumping out longer, polyunsaturated triglycerides.
Polyunsaturated, that sounds like the healthy fats we're told to eat. Fish oil, olive oil, that kind of thing.
Similar chemistry. These are more complex molecules. They have kinks in their chemical chains that keep cell membranes fluid. But more importantly, they are often precursors to signaling molecules. They aren't just sitting there, they are biologically active.
So the worm is remodeling its entire body composition. It's switching from storage mode to action mode.
That's a great way to put it. And when they deleted C32E8.9, that shift stopped cold. The worms reverted to the boring, saturated fat profile, and they lost the longevity.
I'm starting to see the full picture now. The gene is the master switch. It sees the stress, it tells the metabolism to start making these high-performance fats, and those fats presumably help fuel or signal the immune system.
That is the hypothesis, the immunometabolic link. You can't have the immune boost without the lipid support.
It's a system-wide overhaul. It's not just patching a hole in the roof.
No, it's reinforcing the foundation. Which brings us to the final piece of evidence, the sufficiency test.
Right, because in science, it's one thing to say a gene is necessary, you take it away, the effect breaks. But is it sufficient? Can it do the job on its own?
Exactly. So the researchers ask, what if we don't break the engine? What if we just give the worm more of the C32E8.9 protein?
They skipped the workout and just took the supplement.
In a manner of speaking, they genetically engineered worms to overexpress C32E8.9. They even tagged it with GFP green fluorescent protein so they could see it glowing.
A glowing green worm. You have to love biology visuals.
Makes the data very hard to miss. And the result was stunning. Even without inhibiting the mitochondria, even without that initial stress signal, just having high levels of C32E8.9 was enough to extend their lifespan.
Wow, so just turning up the volume on this one gene triggered the benefits?
It did. It extended lifespan, and it improved resistance to that nasty bacteria. Now, it wasn't quite as powerful as the full mitochondrial treatment, so there are likely other factors at play, but it was significant.
That confirms it as a central driver. It's not just a bystander.
No, it's capable of initiating this protective program all by itself. It suggests that C32E8.9 is a bottleneck. Normally, it's kept at low levels. Stress releases the brake, levels go up, and the protective program starts.
This leads to the big question, the so what for everyone listening who isn't a nematode. We have the ortholog, ECHDC1. What does this mean for human aging?
It really shifts the paradigm. We have spent so much time thinking about antioxidants or protein folding or just, repairing damage. This paper suggests we should be looking at the intersection of our metabolism and our immune system, the immunometabolic model.
Living longer might be about having a metabolism that supports a highly alert, responsive immune system.
Exactly. As we age, our mitochondria naturally degrade. We know this. They get less efficient.
Just a fact of getting older.
And technically, that degradation should be triggering this protective response. The stress is there. But in aging humans, we get frail. We get sick. We don't turn into super soldiers.
So maybe the signal is getting lost.
That's the theory. Maybe the switch, our ECHDC1 gene, isn't flicking on properly in old age. Or maybe our fat metabolism is so deranged by modern diets that we can't make those beneficial fats even if we wanted to.
That is a sobering thought that we might be blocking our own longevity pathways.
But it's also hopeful. If we can understand how to safely activate this pathway, how to trigger that lipid shift and immune boost without actually damaging our mitochondria, that could be a huge therapeutic target.
Instead of just treating the disease, we treat the system's ability to defend itself.
We reprogram the body to be in that high alert state, but safely.
It really highlights how interconnected everything is. You can't just look at an immune cell in a dish.
No. You have to know what kind of fat the liver is processing. You have to know what the mitochondria are doing. It's systems biology at its finest.
All from a worm that wouldn't die when it was supposed to.
It's amazing.
So, just to recap the journey, the old dogma was stress the mitochondria, get a protein cleanup, live longer.
And the new reality is stress the mitochondria, trigger C32E 8.9, which remodels your fats and supercharges your immune system via SMA4.
And that remodeling, that shift to action mode, is what actually keeps you alive.
And keeps you moving. Remember, these worms weren't just alive, they were active. They were avoiding bad food. They were living life.
It's amazing. I love when a study just comes along and says, nope, try again, to the textbooks.
Keeps us humble. There's always more to learn.
I want to leave the listener with a thought here. We talked about this gene acting as a switch. If a single gene can coordinate such a massive change, from the shape of your fats to your instinct to avoid bad food, it makes you wonder about the signal noise in our own bodies.
It raises a really provocative question, doesn't it? If our metabolism and our immunity are this tightly partnered, how much of aging is actually just a communication breakdown between the two?
You mean if we could just fix the signal?
Like C32E 8.9 does. Could we decouple aging from disease?
Are we dying because we wear out or because we stop listening to our own warning lights?
That's the question I'll be thinking about all week.
Something to mull over, maybe while you're eating some polyunsaturated fats.
Couldn't hurt. A little olive oil, some walnuts. Feed the pathway.
Thank you so much for joining us on this deep dive into the microscopic world of the worm. It is amazing what these little guys can teach us.
It really is.
And hey, if you enjoyed this, if you learned something new today, please do us that huge favor. Subscribe, follow, rate the show wherever you are listening to the Worm Podcast. It helps other curious minds find us.
And we'll catch you on the next deep dive.
Thanks for listening.